All right, then, well, I just thought I would introduce Veronica. Veronica's got extensive experience in microbiology, having completed a BSc and a master's in microbiology in her home country of Nigeria. And that was followed by 10 years of experience in a variety of labs to do with the microbiology of and disease of aquaculture systems. She joined my lab in 2018, and in her time in the lab has undertaken some genomic analysis of sequence data on a large number of E. coli strains, as well as some experimental work. And so she'll tell us about her PhD research in the next hour or so.
Thank you. Fantastic. You're done?
Yep. So Veronica.
Thanks. Sharon, can we get the screen bigger again? Sorry. Thanks, David.
Can we try to do it?
I just get the screen so Veronica can see it properly. Oh, like that.
Or you want this side, Veronica? What
Yeah.
works for you? All right. How do you want it? Yeah. So you just
Yeah,
press
fantastic.
to advance.
Okay, thank you.
Thank you.
So thank you, everyone. My thesis and research topic is why do some human-associated E. coli strains lack antibiotic resistance? E. coli is a rod-shaped gram-negative bacterium that exists in the as commensals in intestines of humans and animals. Most strains of E. coli are harmless, but some strains, when opportunities arise, can become pathogenic. These pathogenic strains are subdivided into intestinal pathogenic strains and extra-intestinal pathogenic strains. The intestinal pathogenic strains are those that cause diseases like dysentery in the intestine, while the extra-intestinal pathogenic strains are those that colonize sites outside the intestinal system, and they cause diseases such as bloodstream infections, neonatal meningitis, and pneumonia, and so on. E. coli is also subdivided into phylogroups with major representatives A, B1, B2, and D. The focus of my research is on the B2 phylogroup. In excess of 8,000 sequence types have been identified, yet in humans, less than 12 are regularly observed, and just 4 STs account for the majority of sequence types from humans, and they are ST73, 95, and 131 from phylogroup B2 and ST69 from phylogroup D. The STs from phylogroup B2, several studies have shown that they are globally pathogenic. And these 3 sequence types each represent about 10% of E. coli that are recovered from humans. The 3 of them also are equally likely to be responsible for extra-intestinal infection. Given how ubiquitous these strains are, it is expected that they all should be exposed to antimicrobials at about the same rate. But several studies have shown that ST131 is highly multi-drug resistance, while STs 95 and 73 are largely antibiotic susceptible. And so this led to the research question: why are the antimicrobial profiles of these 3 lineages, why are there differences in the antimicrobial profile of these 3 lineages? To be able to answer this question, we looked at what plasmids are. We know that most antimicrobial resistance are due to genes that are born on plasmids. Plasmids are double-stranded molecules that are found naturally in bacteria, where they exist as independently of the chromosome and they are self-replicating. These plasmids can be transferred among lineages through conjugation and mobilization. We now decided to examine the plasmid content of these 3 lineages to be able to see if we can answer the research question. In doing this, we outline 3 research objectives. The first one is characterization of the differences in the genomic diversity of the plasmids in the B2 lineage using various bioinformative tools. The second one is plasmid host dynamics. Here, we use 4 different methodologies to investigate the rate of plasmid transfer to K12 hosts. We also carried out genome-wide association study to examine the reasons for the variation in rates of transfer. And we also assess plasmid stability in K12 hosts. And finally, we assess the cost of plasmid carriage to K12 hosts. Then the last objective, we carried out the genome analysis of the 2 common plasmids that are shared among the sequence types. For the first research objective, characterization of the differences in the genomic diversity of the plasmids in the phylogroup B2 lineage using various bioinformative tools. Draft whole genome sequence data of E. coli isolate from blood, urine, and feces taken from people living in Canberra were utilized. 88 strains of SC73, 77 strains of SC131, and 101 strains of SC95 were used. 3 recently developed bioinformative tools designed to detect contigs associated with plasmid DNA were used. They are MyPlasmid, Plascope, and MobSuit. MyPlasmid uses pentamere frequencies. This is a binary classifier that assigns each contig to either plasmid or chromosome using a probability threshold argument. The second application to Plascope is based on a centrifuge software tool that uses a metagenomic classifier and custom database. It assigns sequences based on exact match against the database, and then sorts them into classes, either plasmid or chromosome. Then the last application to the MobSuit uses a reference database approach. It identifies contigs of plasmid origin and then aggregates the plasmid contigs into groups based on an internal clustering scheme by estimating the genomic distance for each plasmid contained in the plasmid database. This is how it looks like. So for the 3, when we use the 3 application tools, if the 2 of them So this is how it goes. So if the three of them says that the contig is plasmid derived, then we take it as a plasmid. But if 2 of them states that the contig is chromosome derived, then we take it as chromosome and not a plasmid. Or if the 2 of them, none of them, they are not agreeing that it's actually contig is a plasmid derived, then we don't take it as plasmid derived. After we have done this, we now, to confirm the results, we also uploaded the assembly genome into a basic local alignment search tool. This is to be able to search out the basic local search tool. So we uploaded the assembly genomes in basic local search tool to confirm the result that we got from the first analysis that we did. Then the assembly genomes were also uploaded into the Center for Genomic Epidemiology, where there are several tools. There, the plasmid finder was used to find the type of plasmid that is in the assembly genomes. Then the PMLST, also known as F plasmid sequence type, was used to identify the similar fertility plasmid in the assembly genomes. The RES finder was used to identify the resistant genes in the assembly genomes. Serotype finder was used to find the serotype. The FinH typer was used to find the FinH type. The virulence finder was used to identify the virulence genes. And also the MAXI finder and the ConjuScan. The MAXI finder was used to identify micromolecular compounds and the genetic pathways in the protein database. And the ConjuScan was used to identify the conjugation systems in the assembly genomes. The integron finder was used to identify integrons in the assembly genomes. Integrons are genetic elements that capture express and transfer genetic resistant genes using their encoded gene cassettes. So statistical analysis of this data were done, and this is the results. The first result shows the number of plasmid per strain with respect to ST membership. Here we discovered that there was no significant difference in the number of plasmid in these 3 lineages. However, we observed that most of the strains of the SC lineages have at least 3 plasmids, while there are some strains of these 3 lineages that have about 11 plasmids. Here, we discovered that the plasmid DNA content among the SC lineages differs. SC73 lineage has an average of 112 kb, while SC95 has about 164 kb, and ST131 has about 147 kb. We also discovered here that most of the plasmids of ST73 are less than 40 kb. And these plasmids are actually mobilizable plasmids that cannot transfer on their own, unlike the plasmid that we are seeing in ST95 and ST131, which are much more plasmid DNA content, are much more bigger and are more of conjugative plasmid. Here we looked at the incompatibility groups in the ST lineages. Here we also discovered that about 29% of the strains of SC73 do not have any incompatibility group, don't belong to any incompatibility group at all. But then in F1b and in F2 were common in the strains of the 3 lineages, while in F1a was common was prevalent in the strain of ST131. We also found some plasmid that are mobilizable, and the core plasmid was also common in the strains of the 3 lineages. The conjugation systems in the 3 lineages, we also discovered that most of the strains of the 3 lineages have at least 3 conjugation systems, but conjugation system F was common in the 3 lineages, while conjugation system G was only found in the ST95 lineage. Here also, we looked at the F plasmid sequence type diversity in the 3 lineages. F plasmid sequence type diversity, as I said earlier, is a classification type for similar fertility group plasmid. And so here we saw diversity in the F plasmid sequence type in these 3 lineages. But then we observed that F51AB10 was only found in the strains of SC73 and ST95. Why F29AB10 was shared among the strains of the 3 lineages. The other F plasmid sequence types that were discovered is either they are only found in a particular lineage, or they are very small. Their number is very small in each of the lineage where they are found. Then distribution of integrons, positive strains, also described that there was no significant difference in the number of integrons in the 3 lineages. However, when we examine the integron content, integrons that are associated with the F plasmid sequence type, we discovered that F51AB10 harbored about 64% of integrons, while about 88% of F29AB10 plasmid do not harbor any integron at all. We also discovered that some of the other F plasmid sequence type here is that the integron content is very small, or they do not have at all, like the FAB sequence that does not have any integron at all. Distribution of the resistant gene with or without integron. We also discovered here that ST131 strains that have integrons were also associated with many resistant genes. This was followed by ST95 and then ST73. Plasmid encoded virulent genes per strain were also discovered in these lineages, and they were distributed among the lineages. For example, the cytotoxic secretizing factor 1, which is common with patients with urinary tract infections, was common in the strains of SC73 compared with the other sequence types. We also saw some adhesion proteins that are also either common among them or differences in the STs. There's also iron that is common with SC73 compared with the other STs. And we also have the resistance, a complement resistant protein, TRAT, that's also common among the 3 lineages. We also have plasmid encoded by tyrosine that is also common among these 3 lineages, especially colicin E1 and colicin V, which is common with the 3 lineages. However, even though they were seen in just few strains of these lineages. Then we also have the colicin IA and colicin IB that is only found in some strains of SC131 and ST95, while microcin B17 was found in just about 5% of ST131. These bacteriocins are very important because they are responsible for cytolysis and defense responses in cells. Conclusion: SC73 is less likely to carry conjugative plasmids. SC131 strains have integrons that encode a greater number of antibiotic-resistant determinants. Strains with F51AB10 encode many integrons. Distinct genetic diversity was observed in the plasmids of the phylogroup B2 lineages. Now that we have established that there's a distinct diversity of the genes in the plasmid of this phylogroup B2 lineages, we now decided to look at the host plasmid dynamics. In doing this, the first thing we did, the first methodology, we investigated the rate of plasmid transfer to K12 hosts. The strain collection, we used 180 E. coli strains belonging to a 3 phylogroup B2 lineages. And antimicrobials in this 180 E. coli strains are E. coli strains that are selected that are conjugative plasmids. I've been selected to conjugate, they have conjugative plasmids because they are the only one that can transfer on their own. Then in the antimicrobial susceptibility testing, we selected for strains that are resistant to either ampicillin or tetracycline. Then the K12 host is our recipient. The recipient is a plasmid-free, lactose-free, lactose-negative E. coli K12 host that is resistant to nalidixic acid. Then the rate of the plasmid mating experiment was conducted, and the rate of plasmid transfer was calculated. In calculating it, we estimated the density of the transconjugal strains divided by the multiplication of the donor cell with the recipient cell. In the laboratory, this is how it was actually done. Overnight, growth colonies of the plasmid-free strain and the conjugative plasmid-bearing conjugative strain were inoculated into 10 ml lysogeny broths. Then after 24 hours growth, 100 microliter of each of them were added together in a fresh 10 ml lysogeny broth. And they were incubated in a shaker at 150 revolution per minute for 24 hours at 35 degrees centigrade. After 24 hours, 10 microliter of these cultures were serially diluted. Now the diluent, 100 microliter of the diluent were spread to already prepared tetrazolium chloride lactose plates. that has been incorporated with ampicillin for donor cells and nalidic acid for recipient cell, or the combination of the two antibiotics for transconjugants, and they were also incubated. So after 24 hours, this is how the plates look like. So the plates that have the ampicillin, that have been incorporated with ampicillin, only the donor cells will grow. The plates that have been incorporated with nalidic acid, only the recipient cell, the K12 cell strain will grow. While the plates that have been incorporated with the two antibiotics, the transconjugal cells will grow. And the colonies were counted, and they were the rate of transfer was calculated, as I explained earlier, and statistically, this is what we observed. We observed that the rate of plasmid transfer by lineage, there was no significant difference in the rate of plasmid transfer by lineage. So what this is telling us is that the host genetic background is not influencing the rate of plasmid transfer. But then we noticed that there were some small differences in the rate of plasmid transfer by F plasmid sequence type. We now decided to focus on the F plasmid sequence type. And in doing this, we go to the second methodology. Here we use genome-wide association studies to find out the genetic determinant of variation in the rate of plasmid transfer. In doing this, we also use some application tools. PROCA is an application tool which was used to assembly draft, where the assembly draft genomes were annotated. Then we use the RUARI, also an application tool for the pangenome analysis. So the RUARI produces clusters of similar gene groups. So it produces a matrix for the presence or absence of variable genes. So a random forest method using variable gene content data was now used to predict the variation in the rate of plasmid transfer. After this analysis, these were the genes some of the genes that we discovered. So the conjugate transfer protein, TRA-H, TRA-R, conjugate pilus assembly protein, TRA-F, so the conjugate transfer protein, TRA-R, type 4 secretion system protein, TRA-C, and so on. It's because that most of these TRA genes are actually found in TRA-operon. So TRA-operon is a genetic regulatory system where genes coding for functionally related proteins are arrayed. The F plasmid TRA-operon encodes most of the proteins that are required for bacterial conjugation. Approximately 30 kb TRA-operon encodes about 40 genes, and each of these genes encodes for a different protein product, even though not all of them are essential for conjugation. We now decided to look at the distribution of these TRA genes in the key F plasmid sequence type, and we discovered that majority of these TRA genes, in fact, all of these TRA genes were actually more in the F plasmid sequence type F29AB10 and the F51AB10, which is common in only this, which is common in ST131 and ST73 and ST95. We also now decided to look at the effect of these TRA genes on plasmid transfer, and we discovered that even though all the plasmids were able to transfer in the absence of the multiple TRA genes, but the presence of the multiple TRA genes enhanced positively the rate of plasmid transfer. Conclusion: there is no variation in the rate of transfer of this plasmid with respect to the lineage of the bacterial host. However, within the lineages F plasmid sequence type, there were some differences, and most of these differences can be explained by the presence or absence of genes in the TRA-operon. Now that we have established that This F plasmid sequence type are also have an effect in the rate of plasmid transfer based on the trigen that they are encoding. And these F plasmid sequence that are all associated with the plasmid in these strains. We now decided to assess how well the plasmids are maintained within the host using stability experiments. In doing this, 60 transconjugate E. coli strains derived from conjugate transfer of donor and donor E. coli strain bearing plasmids were used. Antimicrobial susceptibility testing of the donor E. coli strain and transconjugate also carried out, as I explained earlier, and the plasmid loss measurement was done. At the end of the experiment, stability frequency was calculated by estimating the log 10 of the densities of the bacterial cells containing the plasmid divided by the densities of all the bacterial cells in the medium. This is also how it was done in the laboratory. So single colonies of transconjugate strains after 24 hours incubation using lysogeny, after 24 hours strain were inoculated into 10 ml lysogeny broth without antibiotics. They were incubated at 150 revolutions per minute at 35 degrees centigrade. Then after the 24 hours growth, 100 microliter of each of these, they were all done in replicates. So after 24 hours growth, 100 microliter of each of them were transferred into a new LB broth plate. And this was also incubated again for another 24 hours. And this procedure was carried out for 7 days. After 7 days, the 10 microliter of the last growth was also severely diluted, as I explained earlier. And they were, the diluents were spread on already prepared rhizolium chloride lactose plates. They were incubated. And after the incubation, we also counted the colonies. And this is what we saw after the incubation. So we discovered that the plates with ampicillin are those that have lost their plasmid, while the plates without ampicillin are the plates that have the cells without a plasmid. Then they were counted. The colonies were counted and were calculated from the way I explained earlier, stability frequency, and then statistically, this is also the result. So here we discovered that some of the strains, some of the plasmids of FC131 were unstably inherited. But generally, there was a significant difference in the plasmid stability rate by the ST lineages. We also observed that there was also significant difference in the plasmid stability rate by F plasmid sequence type. Conclusion. The study revealed that there was host genetic background and plasmid encoded F plasmid sequence type effect in the plasmid stability. We now decided to look at, since the host is carrying the plasmid, is there any effect of carrying the plasmid by the host? And so we examined the fitness scores of the plasmid carriage over time using competition. Using competition experiments. In doing this, 60 transconjugate E. coli strains derived from conjugate transfers of donor E. coli strains bearing plasmid and the plasmid-free E. coli K12 strain were used. At the end of the experiment, the host's relative fitness was calculated using that formula. And the cell density of the transconjugate at time 0 and at 24 hours, as well as the cell density of the recipient at time 0 and at 24 hours, were used for the calculation, for the estimation. This is also how it was carried out in the lab. So in the laboratory, colonies, 24-hour growth of colonies of the transconjugate strain, as well as the plasmid-free K12 strain, were inoculated into 10 ml Lorel broth. They were incubated, separately. They were not incubated at 24 hours, for 24 hours at 35 degrees centigrade and 150 revolution per minute, just as I explained earlier. Then after the growth, 100 microliter of each of those flags were also inoculated into, added together in a new 10 mil red blood flags, and they were incubated. So at this point, we calculated calculated the density of the transconjugant as well as the density of the plasmid free strain. That's at time 0. In calculating the density, we still went back to do what I explained earlier. 10 microliter of the culture were serially diluted. They were now added to already prepared plates. They were incubated, and we counted the densities and we calculated it. But this one, at this point, after we have done that, at this point, we also allowed it to, we incubated it for 24 hours. And after 24 hours, the same procedure was carried out. We collected from it, we serially diluted, and also spread them on the plate and calculated the density at 24 hours. And with this, we're able to estimate the relative fitness of the host. And statistically, this is what we got. We discovered that there was a fairly small significance in the cost effect of the plasmid on the host, but we saw a major significance, a significant difference in the F plasmid sequence type effects on the host fitness. Conclusion. Our studies suggest that there was S plasmid sequence type effect on fitness hosts associated with plasmid carriage. So at this point, we have established that F plasmid sequence type play major roles in the conjugate gene transfer, the plasmid stability, as well as the host fitness. And two major F plasmid sequence types are actually responsible for this. They are the F51AB10 and F29AB10 plasmid. Based on that, we now decided to carry out the genome analysis of the two common plasmids shared among the STs. In doing this, 27 long-read nanospore space assemblies derived from analysis of the genomes of the 3 lineages were used. And also several bioinformative tools were also used. We used Bandage, an bioinformative application tool for navigating the de novo assembly graph. We used to visualize links between assembly contexts. BLAST is also used to examine the regions of similarity among biological sequences. Then RAS is used to annotate or reannotate the genomes of bacteria, while the MAP system was used to construct multiple genome alignment of sequence genomes. We also carry out phylogenetic analysis of the sequence assemblies using the film software. And finally, we analyze the gene content of the 2 key F plasmid sequence type. And this is our result. So the phylogenetic relationship of the assembly SC genomes, we discovered that there were 2 different gene clusters in the analysis. So the first gene cluster is showing genes that are associated with F29. These are associated with F51AB10 plasmid. While the down cluster, gene clusters are plasmid that are associated with F29AB10 plasmid. So this is showing us that the F plasmid sequence type is actually what is playing out in these plasmids. Here also, the map analysis showing the plasmids of F29AB10. We also discovered that there was a very high, that the structures of this plasmid were highly homologous. And we also discovered here that for the plasmids of F29AB10, the resistant genes that are found on them is it that there is no resistant gene or the resistant genes are very diverse? When we look at the plasmids of F51AB10, we saw that the structures were also homologous, highly homologous, but the resistant genes are found in them are similar. We now decided to look at to examine the plasmids of the two F plasmid sequence type. And in doing this, we discovered that in a situation where there is the same resistant gene in the two different plasmids, they are always found in the same contig in the two different plasmids. For example, in this example, a beta-lactamase gene was found in the plasmid of F51AB10 and F29AB10. But when we now discovered that this beta-lactamase gene was actually found in the same contig in the two different plasmids. Here also, we looked at the genes that connected the subsystems and the distribution in the different categories of most of the 29AB10 plasmid types. And we discovered, the subsystems are sets of systems that are connected to functional roles that are connected to specific genes in specific genomes. So here we discovered that for most F29AB10 plasmid type, there is a membrane transport, high number of membrane transport. The membrane transport utilizes the type 4 secretion system in plasmid, which is majorly necessary for bacterial conjugation. And we also have the RNA metabolism and also ion acquisition and metabolism. There is subsystem features that are found in most of the plasmid of F29AB10. There also, we discovered that plasmid coding, protein coding genes were about 29% in most of the plasmids of F29AB10. When we looked at the genes that are connected to most of the F51AB10 plasmid, almost the same thing were playing out, except that there were additional features in the subsystem. The additional systems are like the presence of virulence genes, presence of integrons, and also regulation and cell signaling feature. When we look at the distribution of genes within the F plasmid sequence type, we discovered that there were several trigenes. In this in the two F plasmid sequence type. Like tri-I, tri-Q, tri-C, tri-N, tri-A, tri-D were very common in the two F plasmid sequence type. And these trigenes, as I said earlier, they play numerous numerous roles in conjugal transfer and bacterial conjugation. But then we also discovered that there were some significant differences in some of the genes in these two plasmid types, like the tri-V, tri-J, tri-N, and tri-Y. Even though they are trigenes, but they were more prevalent in the plasmids of F29AB10. Then we looked at the apart from that, we also have the fertility inhibition protein, and also the PAM, the plasmid segregation protein, is also common in the F29AB10 plasmid. But then we also have another plasmid partition protein that is common, that is not found at all in the plasmids of F29AB10. Besides this, we also have some toxin antitoxin systems that are common or different in the two F plasmid sequence type. Like the Hub B is a toxin that is found more in the plasmid of F29AB10, while PEM was found more in the plasmid of F51AB10. Beside it, we have the CCDA and CCDB, which are toxin ionolysis systems that are found, that are more prevalent in the plasmids of F29AB10. We also have another toxin, CHBP, that is found, that is common in the plasmid of F51AB10. Now, these toxin antitoxin systems are very necessary because they help in plasmid stabilization, propagation, and maintenance. So they are very important in this F plasmid sequence type. There are some other genes that are also present, like the alpha gene, which is only found in the plasmid of F51AB10. These ones are also very necessary because they are used for adherence to cell surfaces. We also have the VIB and VIF genes that are not found at all in the F29AB10. These are help to regulate plasmid encoded virulence genes. We also have the in SE1 gene, which is also found only in the plasmid of F51AB10, which is necessary for transposition of insertion sequence ISE. So from this, we can see what is actually happening in the during the plasmid stability and the host fitness. Because of the presence of some of these genes is what is actually we're seeing in the analysis that we did in the lab, in the plasmid stability and the host fitness that we explained earlier. Conclusion: data from long-read sequencing is crucial when investigating the evolutionary relationship existing among different lineages. This study shows that the two F plasmid sequence types common in the strains of the three lineages differ in their plasmid characteristics and genome content. The diversities observed and explained by the differences in the encoded genetic element and the host genetic background. But then, despite these differences, the encoded genes play multifunctional roles in the plasmid maintenance and host cell adaptation and evolution. General conclusion: plasmid associated with specific F plasmid sequence type displays genotypic characteristics that is indicative of adaptation, pathogenesis, and persistence in SPEC diseases. The distinct functions of the trigenes encoded by these plasmids in conjugate transfer is pivotal in the adaptation, evolution, and survival of E. coli lineages. The effects of this F plasmid sequence type on plasmid stability and host fitness play important roles on the ability of the plasmid to associate with new bacterial hosts and consequently on the evolution of plasmid-mediated antibiotic resistance. Study limitations. So some of the strains of SC131 are fluoronolone resistant. But we're not able to look at the resistance of these, so why they are not common in SC95 and SC73. And so we also will have wanted to check if this resistance poses a great fitness cause on SC95 and SC73. So going forward, the FQ resistant isolate can further be selected to test the fitness of the mutant against the parent strain. We also did not consider the activities of phages in conjugate transfer because during bacterial conjugation, a bacteriophage can infect and replicate within the bacterium. So this is often possible because some phages use the conjugated type 4 secretion system as an attachment site for invading the bacterial cell. So their presence is very necessary to prevent. It has been suggested that they can actually prevent plasmid-mediated spread of antibiotic resistance. So going forward, bacteriophage interaction in plasmid transfer should be included in future studies. So finally, I want to thank Australian National University for giving me the opportunity through the scholarship, to carry out my PhD. I want to thank my supervisor, Professor David Gordon, for his objective analysis of my work at every stage. and also his constructive criticisms of my work and my write-ups. I'm highly grateful for all his efforts towards my PhD. I'm also thanking my co-supervisors, Professor Paul Pavli, Dr. Claire, for their constructive criticisms and also how their general contribution to my PhD. I'm very grateful. I'm also thanking the HDR convener, Professor Celeste, for your encouraging words and all your contribution towards my PhD. I thank the E&E heads, Professor Greg and Michael, for making us to feel at home. I thank the technical team, Jack Egerson, Wesley, that have helped me in so many ways. Every time I go to them, they're always there to help me. I thank Adru, the administrator. She has always been there for me. I thank the IT team, RSB, that have always been there whenever I go, those that are no longer in ANU and those that are still there. I thank them for everything that they did. I'm very grateful. I thank my lab and office mates, those that have graduated, Judith, Ma, Truk, Russell, Angel, every one of them. I'm very grateful. Thank you so much for being such wonderful lab mates. I thank all the E&E students and staff who one way or the other has helped me throughout this journey. I'm very grateful. I thank my family, my children, my husband for their patience and understanding for these almost 4 years of my PhD. And finally, I want to thank every one of you that are here to listen to my talk, for me to give my final, talk about my work. Thank you for bringing out your time to come to listen to me. I'm really very grateful. Thank you very much.
I don't know.